Thank you for the kind introduction. Hello and welcome everyone. My name is Christoph Herbel and I would like to start thanking the organizers for this opportunity to present our latest publication to you. Today I would like to discuss with you the preclinical evaluation of novel folate receptor 1 directed CAR-Ts for ovarian cancer via spatial biology, published in Cancers. As just introduced, my name is Christoph Herbel and I'm a group lead in the R&D department of Miltenyi Biotec, working for Miltenyi Biotec since 2017. Let me guide you through today's agenda. I would like to begin with a brief introduction to ovarian cancer and explain you the high unmet medical need in this disease. Additionally, I will give an introduction to chimeric antigen receptor T cells, or short CAR-T cells. We will continue with an overview of our Maxima imaging platform, and I will describe you how we use the Maxima imaging platform in our target discovery workflow. Thereafter, I will guide you through the CAR lead discovery phase, which we performed to identify and nominate a lead candidate. Subsequently, I will present you preclinical data of lead candidate assessment. After that, I will conclude and give you an outlook for future developments. So let's get started with a brief introduction to ovarian cancer. Ovarian cancer is a heterogeneous disease and the main cause remains unknown. It can be divided into 5 histological subtypes. The most frequent subtype is high-grade serous ovarian cancer. At the moment, no efficient screening method is available, and the age of diagnosis is 63. Ovarian cancer is the 7th most common cancer in women, and it is the 8th most common cause of cancer death globally in women. Each year, 300,000 women are newly diagnosed with ovarian cancer. The incidence rate is expected to increase to more than 400,000 in 2040. Every year, 200,000 patients die of ovarian cancer, and forecasts predict an increasing trend to more than 300,000 cancer-related deaths in 2040. The high mortality rate is related to the common diagnosis of advanced stage disease, which has a bad prognosis. This includes relapse rates of more than 70% of patients, and the 5-year survival is only 50%. Currently, standard of care in first-line treatment is a combination of debulking surgery with platinum-based chemotherapy. This regimen is followed by targeted maintenance therapy with angiogenesis or PARP inhibitors. Taken together, a high unmet medical need exists for alternative treatment options for patients with advanced stage disease as well as recurrent disease. A potential opportunity for new and targeted therapeutic approaches are CAR-T cells. CAR-T cell therapy has been very successful in liquid cancers. However, solid tumors are the new frontier in CAR-T therapy. The generation of CAR-T cells is a multi-step process. It starts with the isolation of T cells from peripheral blood. Next, T cells are transduced and expanded ex vivo. And after ex vivo cultivation, the final CAR-T cell product is reinfused to the patient where the CAR-T cells will detect and destroy cancer cells in a target-dependent manner. CARs are similar to regular T cell receptors in combining the TCR signaling in a single molecule. A major advantage of CAR over TCR is its MHC-independent antigen recognition. Thereby, tumor immune escape mechanisms can be overcome. CAR designs have evolved over the past from 1st to 5th generation constructs. Particularly, the addition of further genetic modifications to T cells may help to overcome an immune-suppressive tumor microenvironment. New challenges, such as the tumor microenvironment and accessibility to the tumor, in combination with previous issues like tumor heterogeneity, will ultimately affect CAR-T cell therapy efficacy, and it will potentially raise further or novel safety concerns. CAR T cells are an immunotherapy approach that uses the power of a patient's immune system to attack tumors. CAR T cell therapy has been very successful in liquid cancers. A critical aspect of CAR T cells is the CAR itself. It is engineered to enable T cells to target a specific antigen in a non-HLA restricted manner. This aspect of the CAR is based on its antigen binding domain, which is frequently a single-chain variable fragment derived from an antibody. A hinge and transmembrane connect the scFv to the intracellular domains, which consists of a co-stimulatory and activating domains. CAR T cells are considered to be living drugs due to their potential to persist in patients. However, efficient and safe CAR approaches rely on good target selection. But how can you identify good targets? Promising targets for ovarian cancer, we decided for spatial multiplexing as a method of choice. We employed the Maxima platform to leverage on the power of multiplexing and spatial information for target selection. With conventional microscopy, you are always facing the issue that you can only analyze a few markers on the same sample, and this is very much limiting the understanding of the complex biology in the sample. Our Maxima platform and the mixed technology have been developed to overcome this limitation. With the Maxima, you can analyze more than 200 markers on the same sample, which will provide you unique insights which have never been possible before. And this is only a platform which is capable of performing such kind of analysis. Even if you do not start with such a high number, which might even be a good idea, you will be equipped for the future, as analysis will for sure become more and more complex. You are most probably wondering how we can achieve such a unique depth in the analysis. We are using a cyclic staining process. This means that we start every cycle with the staining of currently up to 3 markers. Then we perform the image acquisition and finally we erase the signal and the cycle starts again. Staining, imaging, erasing. The crucial step in this process is the erase step, as this allows us to repeat this process as often as we want to, and all other influencing factors will allow us. We have 2 methods to erase the signal. One is bleaching of the fluorochrome, and the other method is the controlled release of fluorochromes using our rear dialysis technology. Both methods are very gentle to the sample. So both methods allow you to go beyond 200 markers, and you can use both methods in the same experiment. Additionally, we also provide you with everything you would need to start with such an experiment right away. The platform includes all products you need for every step in the process. Of course, there is the fully automated Maxima system, which will perform all the steps of the cyclic process for you, so that you only have to put in the sample and the antibodies, and then press the start button. Extremely easy to use and no dedicated operator needed. The Maxima system is also very flexible with regards to sample types which can be analyzed, since in principle you can analyze everything which you can put under a microscope: tissue, cultured adherent cells or suspension cells. Additionally, we have the largest portfolio of pre-tested antibodies for spatial biology, which saves you a lot of time for establishing antibodies, and it offers you the possibility to immediately start with your experiments. What makes this platform very flexible is that you can use any kind of antibody you want to image, as the Maxima platform is an open platform, which is built to facilitate as many of your applications as possible. Since data analysis is the critical key to your results, we have an easy-to-use yet sophisticated solution, the powerful MaxIQ analysis software. The software has been solely developed to analyze such high-plex spatial biology data and provides you with solutions for every step in the analysis workflow. Moreover, we provide you with support. We have different levels of global support from our global 24/5 tech support hotline, over our field service engineers to our field application specialists, which can support you especially, but not only with the first steps towards unique spatial biology data and results. Altogether, this is a unique package which will ensure you a fast and easy way to spatial biology. Imaging and spatial portfolio currently is based on two different pillars. Spatial biology with the Maxima platform, and also 3D imaging or light sheet microscopy with the ultramicroscope Blaze. The Maxima platform, with ultra-high complex imaging of cells or tissues, it is possible to stain hundreds of markers on the same sample. It allows for deep phenotyping while maintaining spatial information of the tissue. The ultramicroscope Blaze can image whole organs or even entire organisms while keeping cellular resolution of very large and multiple samples. It is easy and automated and a fast light sheet microscope. Here you can see the 4 hallmarks of spatial biology and why they can be relevant for your scientific question in order to get the full picture. Sometimes there's a need for multiplexing to fully understand tissue structure and function. Some target application examples for spatial biology shown and described, underlining the broad applicability and potential of spatial technologies to contribute to your scientific work. From basic research to clinical settings. Examples include, amongst others, tissue atlasing, biomarker discovery, drug development, but also patient stratification. Coming to the overview of our target discovery to preclinical evaluation workflow. Target discovery is based on the analysis of primary high-grade serous ovarian cancer samples with the Maxima platform. We screened a library of antibodies for potential CAR targets. We selected several potential targets based on expression intensity and frequency for further characterization. This characterization includes target validation, which means that we assess the expression of the selected target candidates on a variety of healthy human tissues. Again, we employed the Maxima platform to perform the expression analysis. Thereby, we added relevant safety information to the potential targets by assessments of on-target off-tumor expression and further refined potential targets. Here we focused on folate receptor 1 as promising target for subsequent development. Next, we generated a series of CAR constructs with varying designs to target folate receptor 1. The different FOLA1-directed CAR T cells were analyzed in vitro and in vivo for efficacy and specificity to identify a lead candidate. After nomination of a promising lead candidate, we performed several optimization steps on the CAR constructs and the manufacturing process. In order to finalize the preclinical evaluation, we performed additional in vitro and in vivo experiments. Eventually, we want to go for clinical translation of our newly defined CAR candidate. Coming now to the data we have generated to identify new targets and target combinations. Here I'm showing you the screening results of potential CAR targets on ovarian cancer samples. In magenta you can see FOLA1 expression on the majority of tumor samples analyzed. Moreover, you can see expression of the tumor marker EpCAM in cyan, as well as the co-localization of folate receptor 1 of EpCAM. Subsequently, we analyzed off-tumor expression of FOLA1 in healthy tissues, depicted in magenta. As you may appreciate, FOLA1 expression is barely detectable in healthy tissues. In addition, I brought you some more examples where multiplex expression analysis of several targets and target combinations was performed in the same experiment in a time and cost-saving manner. In the first panel of tissues, you can see that the target combinations are very specific with few or no cells co-expressing the marker combinations, which makes these combinations attractive target combinations. In the second panel of tissues, you can also see that the target combinations are very specific, with few or no cells expressing the combinations. Taken together, we could confirm FOLR1 as an interesting target on ovarian cancer, and we were also able to confirm absent or low expression of FOLR1 in healthy tissues. Consequently, we selected FOLR1 as CAR target based on elevated tumor expression and low off-tumor expression. In the next step, we started to design and generate CAR T cells, specifically recognizing FOLR1 as a target. I would like to take a moment to line with you on CAR design, CAR T cell generation, as well as assays and readouts. We generated a series of second-generation CAR T cells, which differed in the scFv sequence and orientation. They also had varying hinge domains. So in total, we generated and tested 8 different CAR constructs encoded by the antiviral particle. The workflow of CAR T-cell engineering begins with the separation of T-cells from peripheral blood. Thereafter, T-cells are activated and subsequently transduced with lentiviral particles. After ex vivo expansion, CAR T-cells can be analyzed by different means, for example by flow cytometry. Additionally, functional in vitro assays can be set up. We use co-culture experiments of CAR T-cells with ovarian cancer cell lines as functional in vitro assays. Here we analyze different parameters, including cytolytic activity, cytokine secretion, and activation marker expression of CAR T-cells. Initially, we analyzed the expression of the different CAR constructs on T-cells and could not detect significant differences. All CAR constructs were expressed at high levels. In our co-culture experiments, we were looking at cytolytic activity and specificity of the different CAR constructs by using FOLR1 proficient of 90 cells as well as FOLR1 deficient of 90 cells respectively. All CARs tested reduced the number of FOLR1 expressing target cells indicated by the decrease of GFP signal from the tumor cells. Most CAR candidates are also target specific since FOLR1 deficient cells are not lyzed, except for candidate F, which reduced the number of target cells target independent. In addition to the assessment of cytolytic activity of the different CARs, we also determined the expression of activation markers on CAR T cells after co-culture with target cells by flow cytometry. The CAR candidates express comparable levels of the activation markers CD25, CD69 as well as 4-1BB. We observed that the induction of activation marker expression was FOLR1 dependent too. After co-cultivation of CAR T cells with cancer cells, we assessed cytokine secretion and expression. Although we could not see differences in granzyme B expression between the candidates, we could see differences in cytokine secretion. We collected the supernatant and determined the concentration of secreted cytokines, namely GM-CSF, interferon gamma, IL-2, and TNF-alpha. Expression and secretion of cytokines was target dependent since we could not detect cytokines after co-culture of CAR T cells with target cells without FOLR1 expression. Candidates A and D secreted more cytokines than candidates E to H. To complement the previous activation marker analysis, we also analyzed exhaustion marker expression of PD-1, LAG-3 and TIM-3 by flow cytometry. Candidates E and F expressed more frequently the combination of PD-1 and LAG-3 as well as the triple combination of exhaustion markers, which may indicate a more exhausted cell state. To characterize the phenotype of CAR T cells, we assessed the composition of the T cells by their subtypes. Candidates E and F differentiated more often into effector memory and central memory phenotype in the absence of FOLR1, This may indicate again an antigen-independent activation of these candidates. Taken together, we selected 4 candidates based on cytolytic activity, antigen specificity, expression of activation exhaustion markers, cytokine secretion, as well as T-cell composition for subsequent in vivo assessment. Before discussing our in vivo data, I would like to introduce to you the CliniMax project platform. We use this system to generate sufficient numbers of CAR T-cells for our mouse study and to use a manufacturing process of CAR T-cells that may be relevant for later clinical development. It offers automated GMP-compliant cell processing in a closed system, from cell separation through cell culture to formulation. It allows cell manipulation via transduction or electroporation, and the starting material can be whole blood, PBMCs, and fresh or frozen leukapheresis. Coming back to our mouse study to assess CAR T-cell functionality in vivo, we set up a subcutaneous xenograft model where we injected 10 to the 7 tumor cells and let the tumor cells engraft for 21 days. Then we applied a single dose of 10 to the 7 CAR T cells intravenously. The study continued for another 21 days and we analyzed tumor growth, CAR T cell persistence in blood and organs. As you can see, all 4 candidates reduced tumor burden after day 9. Candidate A presented with the fastest tumor eradication and reached the lower detection limit after 13 days. Analysis of CAR T-cell persistence in blood, but also bone marrow, revealed that candidates A and E were detectable with higher frequencies. Additionally, CAR T-cells of candidates A and E were more frequently detected within the remaining tumors at the end of the study. Interestingly, we observed elevated levels of human GM-CSF, interferon gamma, and IL-9 7 days post T-cell injection, which were reduced at the end of the study for candidate A. Eventually, we selected candidate A for subsequent optimization due to tumor eradication, persistence, homing to bone marrow and spleen, tumor infiltration, and its cytokine profile. Lead optimization included changes in the CAR vector, for example, removal of the truncated LNGFR reporter gene. To confirm that the optimized CAR construct remained its functionality, we assessed efficacy and specificity in vitro using different ovarian cancer cell lines with varying FOLR1 expression. To investigate whether the FOLR1 directed CAR T cells are specific, we performed co-culture experiments with FOLR1 deficient cells. You may appreciate that the FOLR1 specific CAR T cells shown in magenta do not reduce tumor cell numbers. However, looking at 4 different ovarian cancer cell lines expressing FOLR1, we detected reduction in tumor cell numbers. To complement the in vitro characterization, cytokine secretion was analyzed after co-culture of CAR T cells with ovarian cancer cells. Cytokines such as GM-CSF, interferon gamma, IL-2, and TNF were detectable in the co-culture supernatant of FOLR1 expressing target cells. This indicates the specificity for the FOLR1 CAR. Last but not least, the expression of activation markers was analyzed by flow cytometry, and we confirmed antigen-dependent reduction of CD25, CD69, and 4-1BB expression. Taken together, these data indicate that the change in the vector did not impair efficacy or specificity of the FOLR1 CAR. Despite the promising in vitro and in vivo data that we generated, we anticipated that the nature of a solid cancer, in particular the tumor microenvironment, may pose additional challenges. Therefore, we set up 3D co-culture assays. This included the generation of spheroids from ovarian cancer cells and subsequent co-culture of these three-dimensional structures with CAR T cells. We observed reduction in GFP signal indicating loss of tumor cells, accompanied by infiltration of T cells shown by CD3 stainings. In order to better resolve the 3D structure, we performed the same 3D co-culture assays, but we used the ultramicroscope-based platform, which I introduced to you earlier. This approach allowed us to quantify the number of infiltrating CAR T cells into the spheroid. In addition, we analyzed the depth of infiltration, or in other words, we measured how far the CAR T cells migrated into the 3D spheroid structure. These data showed us that the CAR we developed is capable to infiltrate into three-dimensional structures. Moreover, we assume that the tumor microenvironment poses immune-suppressive features which may hamper CAR T cell function. In order to mimic this scenario, we envisioned an ex vivo assay that should recapitulate the tumor microenvironment present in an ovarian cancer patient. One approach we use to address this issue is to work with patient-derived ascites. Malignant ascites is the abnormal buildup of fluid in the abdomen, which can be observed in advanced stage disease. This fluid contains multiple non-cellular as well as cellular components, including cancer cells, and has been proposed to have an immune-suppressive function. We separated the cellular from the non-cellular fraction and performed co-culture experiments with CAR T cells. One of many combinations that we tested was the use of patient-derived tumor cells with CAR T cells. Additionally, we replaced the regular culture medium with ascites fluid. Thereby, we intended to investigate whether the cellular or non-cellular ascites compartments may inhibit CAR T cell function. Noteworthy, we observed a strong reduction of FOLR1 expressing tumor cells after co-culture. Complementing to the cytolytic activity, we detected activation and exhaustion marker expression on CAR T cells as well as cytokine secretion by CAR T cells. In conclusion, the promising results from the 3D assays and from the assays with patient-derived tumor samples prompted us to investigate CAR T cell functionality in vivo. Similar to the first in vivo study, we used a subcutaneous ovarian cancer xenograft model. 10⁻⁷ tumor cells were subcutaneously injected and engrafted for 3 weeks. A single dose of 10⁻⁷ CAR T cells or mock T cells was given intravenously. Subsequently, we monitored CAR T cell persistence over time, and at the end of the study, we analyzed CAR T cell homing to different organs and infiltration into the tumor. You may appreciate that in the CAR-treated group, we can hardly detect any tumor signal in the mice from day 9 on, in contrast to the control groups. This highlights the efficacy of the anti-FOLR1 CAR. However, mice bearing FOLR1 deficient tumors have measurable tumors in the presence of FOLR1-directed CAR T cells, indicating the specificity of our FOLR1 CAR. Quantification of the tumor signal shows that the FOLR1 CAR treatment in mice with FOLR1 expressing tumor reached a lower detection limit 12 days after CAR injection, whereas different control groups do not respond to the treatment regimen. Additionally, we performed caliper measurements and observed the same effect. Analysis of CAR T cells over time revealed target-dependent expansion and persistence in peripheral blood. Persistence in blood was accompanied by homing to spleen and to a lesser extent to bone marrow. In the remaining tumor pieces, we were also able to detect infiltrating CAR T cells. Taken together, we confirmed the optimized FOLR1 CAR candidate as lead candidate due to tumor eradication, specificity, persistence, homing to bone marrow and spleen, tumor infiltration, cytokine profiles, as well as advanced in vitro assays. With this, I would like to conclude on our target discovery to lead optimization workflow using mixed technology. We identified and confirmed FOLR1 as promising target in ovarian cancer, a devastating disease with high unmet medical need. We analyzed FOLR1 expression over a variety of tissues to validate off-tumor expression. Next, we generated a series of second-generation CAR T cells and selected a lead candidate based on in vitro and in vivo data. Finally, we optimized the lead CAR construct and performed in vitro and in vivo characterization, including advanced in vitro models to confirm efficacy and the specificity of this candidate. Currently, we are in the process of clinical translation. At the end, let's switch gears and imagine how one could further improve CAR constructs. Therefore, I brought an example with me from another study we performed. Here we analyzed xenografts from mice treated with two different CAR constructs, which performed similarly in vitro. However, in vivo functionality differed, and we wondered about the reason. Therefore, we performed spatial multiplexing. In particular, we investigated CAR T-cell infiltration into tumor, tumor remnants, macrophage infiltration, as well as target expression within the tumor. We found one candidate that was not able to infiltrate a tumor. Thus, target-expressing tumor remains, and additionally, murine macrophages were also not able to be recruited by CAR T-cells. This example should highlight that spatial multiplexing may guide the improvement of CAR T-cells and other drugs by shedding light on characterization of disease mechanisms, tissue phenotyping to identify new targets or cell types, identification and characterization of cell-cell interaction and function, revealing the mode of action, investigating drug resistance mechanisms, predicting drug responses via biomarkers, and identification of prognostic biomarkers. I'd like to give you a sneak peek at what we are doing. Here you can see two proof-of-concept datasets where we stain different RNA targets and proteins on the same tissue section using our antibodies and RNA escape probes on the maxima. In the first image you can see a beautiful correlation of two different RNAs with the morphological structure of the sample, non-small cell lung cancer. RNA-Sky is completely developed by Miltenyi Biotec and uses a proprietary technology based on control, flow chrome, release and mix. If you already have a maxima, this technology will be available without any modification to the instrument and only will require the RNA-Sky reagents and a software update. Combining antibodies and RNA probes will further support you in solving your scientific question. Last but not least, there's also the possibility to combine the 3D and multiplexing technologies to gain even deeper insights into the samples of interest. Our workflow combines 3D imaging with multiplex spatial analysis to get an overview of complex large samples like xenografts or patient samples, identify target structures within them, and to further analyze a carefully selected region in depth with hundreds of markers, all on the same valuable specimen. With this, I would like to end and thank you for your attention, and I'm looking forward to your questions.